until you get the final result. So a lot of times you've got a file with a piece of paper attached, and each day the microtech is adding notes to it.
Okay.
For communication among personnel. Because if I did it today and I'm off tomorrow, we gotta have some way to be on the same page.
Yes, sir.
Now this chapter 9, it's about all these biochemical tests. And originally chapter 17 was supposed to be before this, but when you start looking, it starts mentioning a lot of biochemical tests that we have talked about not at all. So we're going to talk about them first. And one of the things I want to do up here first is there's something called KIA and TSI. And they're done in a slant tube. And the agar gel is actually poured in like this. And the way it's set up, you take a needle,
You stab it.
stab straight down, come out, and then you streak this part.
Kind of looks like a Christmas tree.
So being that that's a gel, once you stab it, it's going to close back on. Which means everything from here down is an anaerobic environment. No oxygen. The top is aerobic environment. And that's how we can tell how an organism reacts in two different environments in the same media is these slant tubes. Because we know how it reacts with air and without air. So it's going to mention that, and I wanted to kind of show how that's set up and how it works before. And here's one right here. It's what is this, KIA? Yeah, it's KIA. And it's actually poured in as a liquid on a rack that's slanted and let it set up.
You already buy it like this stuff?
Yeah.
Oh, okay.
You can make your own.
You can make most of it. Take it now, pass it around,
Now, we've mentioned a couple of biochemical tests on gram-positive organisms. This chapter is more specific for the gram-negative,
Okay.
but there are some that cross over. Like we're going to talk about the oxidase test. So there was a gram-positive organism we identified with the oxidase test, Micrococcus. It's a modified oxidase test. They call it the microdase, but it's very similar. But most of the rest of these are going to be more specific for gram-negative organisms. This was the traditional way to identify organisms. The problem with it is, because it works very, very well, is expense. Number two, in order to identify most organisms, you have to have 10, 12, 15 different tests. So you've got a whole rack full of these tubes for one organism. So think if you've got 50, 60 patients, some of them have 2 and 3 organisms, you've got hundreds upon hundreds of racks filled with tubes. And that's why they started getting away from this. The expense of all of these sterile glass tubes that you have to buy, the space requirements, you have to have huge incubators. And that's why they started getting away from this, but most of the methodologies have not changed. One of the tests they do is carbohydrate utilization. The morphology, or the differential characteristics on McConkey, is a biochemical test. It tests for lactose fermentation. If it's pink, they ferment lactose. Lactose is actually a disaccharide. It's made up of 2 sugars, glucose and galactose. Now we said that all bacteria, the sugar they work off of is glucose. So in order to ferment lactose, they have to be able to break this disaccharide to get to that glucose. To do that, they have to have 2 enzymes. Beta-galactoside permease, that's what grabs that lactose and brings it into the cell wall so they can work on it. The other one is beta-galactosidase, which is what actually breaks it, where they can get to that glucose. Then they'll metabolize it through a couple of different pathways. There is something called a delayed lactose ferment, and that's where they're lacking the permease. They can't bring that lactose inside of them so that they can work on it. But what they can do is push this to the outside and break it up externally and then absorb the glucose. They can still do it, but it takes longer. So they're a delayed lactose fermenter. If they've got both of those enzymes, they can do it pretty quickly because they can pull it into the workshop, break it down, use it up, and move on. But if they're missing that first one, they can do it. It's a lot slower. Remember also that all organisms that can ferment lactose can ferment glucose. All organisms, because that's basically what they're breaking it down to. But not all organisms can ferment glucose. And the question at the bottom, I've already answered that. Delayed slow lactose fermenter, they're missing that first enzyme. So they can organisms use two different methods to convert glucose: aerobic and anaerobic, oxidation or fermentation. Oxidation fermentation test. Fermentation produces a very acidic end product. It goes on to say they can actually produce more than one acid, mixed acid fermenters. You don't need to know about that. Just know that fermentation, the end product is very acidic. And that KIA tube that I pass around, if it's fermenting and acidic, it will show up as a color change in there so that we know it's acidic. Oxidation is a much less acidic method, so you won't have that color change. It says the OFBM, OF basal medium, is an example of an oxidation fermentation test. It has a pH indicator built into it. Use two tubes. You seal one of them with mineral wool. That creates an anaerobic environment. So uninoculated, the media is green. If it produces acid, it will turn it yellow. So in this one, the first example, both the sealed and this big blob is the mineral oil that's jellified. Both of them are yellow. It produced acid both ways. It fermented in both environments. The middle one you can see is yellow only in the open tube. So it was not able to create that acidic environment in an anaerobic environment. And this one couldn't utilize it either way. It's a non-fermenter. It couldn't ferment in either environment. Neither one of them changed colors. So this test, the OF medium, is basically which environment are they capable of fermenting. And that's just one clue for the identity of the organism. That's why I said, to say this was our test, this organism we tested, this was the result. Well, there's hundreds of organisms that will give this result. That's why you can't ID based on just one. Yeah?
The original color of this medium is pink.
It's a pinkish, sometimes often green. Yeah, I know it's red. But red, being in a pH indicator media, red indicates alkaline, yellow indicates acid. So this talks about the triple sugar iron order. It is virtually identical to the KIA that I passed around.
Except
Looks the same, works the same, the whole 9 yards. The only difference between the two is that TSI has sucrose in it in addition to glucose and lactose. KIA just has the glucose and lactose. TSI has a third sugar, that's why it's called triple sugar iron. Both of them contain ferrous sulfate, which is an iron derivative. Any organism that produces hydrogen sulfide, or H2S, will turn that ferrous sulfate black. That's a big clue. A lot of our pathogens will produce hydrogen sulfide. So when we see that that media has turned black, that's another clue. We know they produce hydrogen sulfide. It also has a pH indicator. So we're looking for alkaline versus acidic results. If it's yellow, it's acidic. If it's reddish, it's alkaline. So if it produces no fermentation in the bottom of the tube, which is anaerobic, or the top of the tube, which is aerobic, it will be alkaline, or written as KK or alkaline, red over red. The enteric bacteria, which we're talking about your E. coli, your salmonellas, your klebs, and all that, will typically be an alkaline slant with no change at the bottom. One thing I don't think it tells you on here, and it should, if it turns black, you can't see if it's yellow or red. But that reaction will only occur in an acidic environment. So if it's black, it's acidic. In other words, behind that black, it's yellow. You just can't see it. So if you can't see it because it's black, you write it as acid, not alkaline. So glucose fermentation only means it cannot utilize lactose or sucrose. It will be alkaline over acid. The small amount of the glucose in the media is fermented, which will initially turn the whole thing yellow, usually within 8 hours or so. But as soon as it uses up that glucose, it'll start metabolizing the peptones in it, which will turn the top of it back to the reddish color or alkaline. That's why it's very important to read these after 24 hours and not read them early. Because if you read them early, you'll say acid, acid, and that is not true. It will start that way, but that is not our final result. It will go alkaline over acid. If it can utilize the lactose and the sucrose, it'll stay acid over acid. It'll utilize that glucose first, turn the whole thing yellow, but then it'll say, well, I'm out of that, well, let's grab these other sugars, and it'll continue its journey, and it will maintain that acidic environment. So if it stays yellow after 24 hours, the whole thing is yellow, then we know that it can utilize either lactose or sucrose, depending on which one of those two medias we're using, or maybe both. So it can ferment more than just glucose. Again, hydrogen sulfide production, usually an alkaline slant with an acidic butt with H2S. So it would be written K over A with H2S. And we know that the butt of that tube was acidic because it's black. It can't be black unless it's acidic. So even if we can't see the color, if it's black, it's acidic. And it does say the butt may be completely black, completely obscuring any color that you can see. But you know that it has to be acidic. Gas production. This is another big clue for a lot of these gram-negative bacteria. Some of them will produce carbon dioxide gas, some will not. A lot of times you'll see bubbles in that media. A lot of times it'll produce that gas at the bottom, and it'll actually push that entire slant of media up from the bottom of that tube about this far. So there's a gap at the bottom, which tells us they're a gas producer. We still don't know what it is, but that's another clue. So let's say that it was yellow over black, and it was pushed up from the bottom of the media. That would be acid over acid with H2S and gas production. So 4 different clues we've got about this organism off of this one tube. So number 1. Acid over acid with gas. Can you see it back there? Yeah, you should be able to see it where it's pushed up from the bottom.
Yep.
The gas production has built up enough pressure to shove that media up. The top of this is yellow, the bottom of it's yellow, so it's acid over acid. Number 2, this is yellow. We can't see what color this is. It's black.
Because it's black, we can assume that it's yellow underneath. So
Yeah, it has to be, otherwise it could not be black.
We call it acid over acid.
Yes, the answer to that would be A/A with H2S. Now what about number 3?
Oh,
That's gonna be,
looks normal.
alkaline acid.
Yeah, there's yellow at the bottom.
So it's gonna be glucose.
It's a little bit yellowish at the bottom.
Okay.
Yeah,
Reddish on the top?
yes, it's gonna be glucose, so it's alkaline over.
What do you call the top color on?
It's very reddish, it's not yellow like this.
Very red.
The top of that is still very reddish.
Mm-hmm.
I would have to call that alkaline. There's enough yellow at that bottom that I would call that acidic. Because if you look at that last two, that's alkaline over alkaline. See how red that is?
Mm-hmm.
This is yellowed up enough that I would call that acidic. So alkaline over acid. I don't see any gas bubbles. It's not pushed up. It's not black. So no gas, no H2S. So your answer on this one would just be K/A.
Okay.
These are always read top to bottom, by the way. K/A.
Okay.
What about number 4?
A over A with gas.
No.
Tricky.
Wow.
If you look at that very closely, and it's hard to do on this picture, I know. The very top of that slant is reddish. This thing has produced so much hydrogen sulfide that it's tried to discolor this whole thing. But look up here. That's not black. That's red. What else?
Gas.
Gas producer. This won't always happen. A lot of times you'll look and your medium will be split, which, this gas will escape and tear the media and get to the top. Usually when you unscrew the cap, those will spew. Yeah.
Oh.
Is there any difference between labeling it as gas and then hydrogen sulfide? That's gas also.
That's two different. Like number 1 and number 4 both have gas. See how they're pushed up from the bottom of the tube?
Mm-hmm.
Occasionally you'll see it where it's just bubbles in here. Occasionally this gas will try to escape and rip the media. And every time it does that, when you unscrew it, it'll spew. The gas will get out. But most of the time, it'll push it up like this, which makes it a lot easier to read.
Whenever that gas is slide back on, it stays.
Yeah, just stay there. And then number 5, red on top, red on bottom, no gas, no H2S. It's just alkaline over alkaline. And there's actually a chart in your book that tells a lot of different organisms with their GSI results.
Mm-hmm.
So maybe in chapter 19 under the inheritance when you get there, but it's in that book somewhere. Yeah.
Okay, so number 4, there's some emptiness at the bottom.
Yep.
Number 1, there's also some emptiness
Yes.
at the bottom. But because number 4 is black, we can guess that that gas is going to be hydrogen sulfide. Whereas the number 1, because we don't see anything black, we can't really identify it as a specific gas, so that's why we just label it as gas.
Yeah,
right?
we're just gonna label both of them as gas, because we don't know that that's hydrogen, so it could still be CO2 gas.
In number 4.
Yeah,
Because in number 4, we did label it as hydrogen sulfide.
and that should also be comma gas.
Thank you.
That's, I gotta fix that.
Okay.
That should be K/A, comma, H2S, comma, gas.
Thank you.
Yeah, good catch. I should have caught that. Huh?
Does it have a color gas as well?
No, it's just because you said.
Number
the volume's
2
moved
is
up.
not pushed up,
Yeah,
so there's no gas pressure.
Thank you.
There is one huge clue whether or not that's CO2 or hydrogen sulfide gas. If it's escaped when you open it, hydrogen sulfide is especially foul. I mean, it stinks. You open it and it spews and you catch that whiff, you'll know it was hydrogen sulfide gas. But in this, we're not trying to identify the gas, presence or absence thereof.
Sulfides usually smell like rotten eggs, don't they?
Sulfur compounds stink no matter what the compound is. Hydrogen sulfide is one of the gases produced during the decomposition of feces, whether in your bowels or in a sewage treatment plant, and that's what produces a lot of that lovely aroma.
When I was in the 11th grade, you toured the sewage plant, but you just watched this.
Yeah.
And this was a did.
Hello.
Yeah.
So we've been looking at this medium, which is the TSI medium.
Yeah.
Could we see similar results if it was in a KIA medium?
Yes,
Thank you.
but KIA only has 2 sugars instead of the 3. That's the only difference. Carbohydrate utilization. I'm not even gonna try to say that word. You need to remember ONPG. This is the test for the detection of those delayed lactose fermenters that we mentioned. These slow fermenters may appear as non-lactose fermenters on the primary isolation medium, as well as your TSI. So when you look at that 24-hour plate on McConkey, that colony will be clear. You put it back in the incubator and look at it a couple of days, it'll turn pink. We don't want to wait 3 days. We want to catch it quicker than that. So ONPG is structurally similar to lactose, but it's a lot easier for these organisms to transport into inside the cell wall. Does not require that enzyme. So to perform the test, ONPG is typically either a paper disc or a tablet. Looks like a little white aspirin. You'll put your organism that you're trying to test into like a heavy suspension of saline where it's cloudy. And then you add that to the disc or the tablet, whichever the case may be, and incubate it at 37 degrees. Usually within 6 hours, you'll see a positive result. There's a figure in the book that shows you what positive is. Depends on manufacturer. Usually it'll turn purple. That's positive. So if it turns purple, you know that what you have is a delayed lactose fermenter. So if you have that clear colony on McConkey, but you're like, okay, is this truly a non-fermenter or is it just a delayed one? You can do this within 6 hours, you'll know. Versus throwing that plate back in the incubator and reading it in 2 to 3 days.
Okay, so what we'll do, take our McConkey, pull a little colony off of the McConkey.
A clear colony, not pink.
Oh, right. Pull it off and then put it on one of these discs.
No. You'll create a heavy suspension in sterile saline.
Gotcha.
To where it's cloudy. I mean, you want a lot. And you only need a drop. So put that much saline and go to stirring a colony in there until you really, because you got to put it on there with a dropper.
So
So
this
you
is
want
in
it
a,
liquid so it'll soak in.
this is in a tube then.
Yeah, you got it in like a blood bank tube with some saline. And you stir that organism in it.
It's on page 178.
Is that where it is?
Picture on the
Yeah.
right.
Then you put it on there and within 6 hours you'll have your answer usually.
And you gotta take the ONPG disc and put it in there too?
No, you put a drop of your saline suspension onto the disc,
Ah.
then incubate.
Okay.
And remember, they have these delayed lactose fermenters, they have that other enzyme. They can break down lactose, they just can't transport it. That's what makes them slow. Glucose metabolism, it's metabolized into an intermediate byproduct, pyruvic acid, and it's further degraded down one of 2 pathways. Don't worry about those too much. There's a couple of test here it mentions, and it's what they call the MVIC reaction, which is actually a battery of tests. Indole, methyl red, Voges peroxiscour, and citrate. The methyl red test, you know, we mentioned some of these are mixed acid producers. Let's test for that. You add methyl red indicator, positive will turn pink. The Vogue peroxiscour tests for stable, neutral products after the addition of the indicator. And then the methyl red, the VP, we'll talk more about. So you're going to need to know that MVIC reaction, what's in the MVIC. Indole, methyl red, Vogue peroxiscour, citrate. Note at the bottom, organisms tend to be positive for the MR or the VP, but not both. Why? This is testing for mixed acids. This is testing for neutral. You can't be neutral if you produce mixed acids.
So it's gonna be.
You're going to be one or the other. Or you can be negative for both, but you're not going to be positive for both. Yeah.
In Vogue peroxiscour, that neutral product, that neutral refers to its pH, yeah?
Yeah.
Okay.
Neutral pH. So again, if you're doing a battery test, you're doing this MVIC battery, and your MR and your VP are both positive, you gotta do them again. Something's not right. You can't be acidic and neutral. Amino acid utilization. These are important because not all organisms can utilize glucose. Most of them can. So we do tests for amino acid utilization. Decarboxylase test determine whether the organ has enzymes that can remove the carboxyl group. The amino acids lysine and ornithine are most commonly used. And the two medias, MIO and LIA, contain lysine and ornithine, respectively. Deaminase test, whether they can remove an amine group. One of the example is a PAD, phenylalanine deaminase test. Test whether the organism has that deaminase enzyme. Test media contains phenylalanine in slant form. Even though this is a slant, you don't stab this one. You're just going to streak the slant. You're going to inoculate. 24 hours later, you're going to take it out and you're going to add a drop of ferric chloride to that slant. This is a drip, you just let it run down it. A positive result, it'll turn that slant green, which means they have that deaminase enzyme. It goes on to say, Proteus, Morganella, Providencia are positive. We'll discuss those organisms more in chapter 19.
So the Proteus, Morganella, Providencia, they're all gonna cause the green color?
Yes. Okay. They're all going to be, when you add that ferric chloride, they're all going to have that green color. They're going to be a positive result. When you do your group unknown, these tests we're talking about, the way I do that, everybody's going to get a tube or a slant or something, test it, come together with your results, ID the organism. That's why we got to have actually a large enough group to pull that off, or somebody's got to do multiple tubes. Because it takes a multitude of tests before you get all the clues to figure out, okay, here is a most likely result.
We had to do this in micro, and we had to make our own guide to how to distinguish between if it's positive and negative and what organisms.
I'm gonna help you out with that though.
Hey,
So here's the PAD. It's a very nondescript color originally. Kind of a clearish yellowish look. So you're going to streak it. After you add that ferric chloride in the middle, you can see that slant turn that dark green color. That was a positive. That number 3, you just see that dark orange color. That's just the color of the ferric chloride. That's why it has that darker color. Before the addition of the ferric chloride in both of these tubes, it's still going to look like this. It's not going to change color or do anything until you add that ferric chloride.
is this the concept of like what's in the wells whenever you add certain stuff to each well and you're trying to isolate the organism? I know you don't have that certain type of equipment at National Park, you have something different. You have the.
We'll talk about that at the end, but you're very close to, you're in the right ballpark.
Okay.
Miscellaneous tests, citrate utilization. This is another one you'll do on your unknown. Determines if an organism can utilize sodium citrate as its sole source of carbon. Simmons citrate is most commonly used, and that's what we have. It has a pH indicator. If it's positive, it'll change from green to blue. Simple enough. You streak the slant, incubate it. If it turns blue, positive.
And is that part of the MVIC battery?
Yes. DNase test. Take that SE off and think DNA. This tests for enzymes that can cleave the DNA bonds in the medium. Staph aureus and an organism called Serratia marcescens are DNase positive. They can cleave the DNA bonds in that medium. Gelatin liquefaction. Test for gelatinases. Enzymes that break down gelatin into amino acids. So you have this gelatin media, you add the organism, it'll break that media down, so instead of the next day, instead of that real soft jello, you have a liquid. endo production. This is a biggie, done on almost all gram-negative organism studies. Real quick, real easy. Test for the presence of endo, which is a degradation product by the action of tryptophanase on tryptophan. Tryptophan being the protein that makes you sleepy if you eat too much turkey. It's also on a lot of your sleeping medications now. The way they're talking about doing it is correct. There's a new method. The organism is incubated in broth containing tryptophan. After incubation, two methods can be used. Ehrlich's or Kovacs reagent, we use Kovacs. You add Kovacs reagent to the broth, it develops a red color that's positive for that endo production. There's actually a newer method to do this, which is way easier, and that's why it's done so commonly. You take the colony you want to test and pick it up with a swab, take that reagent, drip it onto the swab, and it'll change colors if it's positive. This is one of those tests that's still done, even though we're modernized past a lot of these biochemicals. This one's still done because it's literally drip. Oh, it's positive. It's so quick, so easy. The droppers are so inexpensive. This is done frequently. And it helps us out because when we go and put this organism on our $1 million machine to identify it, we can type in it's endo positive. The machine goes, well, that's another clue to help me identify this, and it will use that information. Motility. Some of these organisms can swim. We talked about Proteus swarming the plate. They swim. Two different ways you can do this. You can wet prep it like a urine. And you can look in there and they'll be wiggling around if they're mobile. The old way is using a semi-solid medium. It's like softer than Jello, more like jelly. And you put it on a needle and stab it straight down and straight out. So when you look at it, you just see a little straight line down the middle of that medium. If it's mobile, when you look at it the next day after incubating, it'll be fuzzy because they're swimming away from that straight line. You'll have a fuzzy line. That lets you know that it's not staying put. This stuff is moving. So it's a mobile organism. Nitrate-nitrite reduction, used to detect the ability of an organism to reduce nitrates to nitrite. Inoculated into a broth containing nitrogen. After 24 hours, you have the reagent, red color is positive. There's a faster method for me. Can't write it down because it's not the right way to do it. When I take a urine and they send it for a UA, and when you open that lid and it whips your head back because it's so bad, 99.9% of the time that is E. coli, and the reason it smells like that because it reduces nitrates to nitrites, and that nitrite is the smell. And it is a very acrid, like burn your nose hairs kind of smell. And it's like immediately, it's like it's nitrite positive. Of course, in micro, we can't say that without testing it. Just like I can sniff a Tricia's plate and I knew it was like an elephant. We would still have to identify that. Oxidase. Another really, really common test that's still in the micro labs because it's so fast, so easy. Used to determine the presence of cytochrome oxidase. You can drop the reagent onto filter paper and rub the colony onto it. You can drop it directly onto the plate, or you can do like the endo, swab it, put your reagent on the swab, and look for the color change. It's literally that quick to do this test. That's why we still do it. Useful in distinguishing the enteric organisms from the pseudomonads. Pseudomonas, the fruit-smelling green one. Mm-hmm. It's also useful in identifying Neisseria, because Neisseria is oxidase positive. When we're talking about Neisseria, we're mainly concerned about Neisseria gonorrhea. So the usefulness of this, when we do a vaginal culture on someone, we're curious, do they have gonorrhea? We'll take that swab directly through that heaviest growth quadrant off that plate. And we'll do an oxidase on that swab. As long as it's negative, We don't know what's there, but we know it's not gonorrhea. If gonorrhea is there, it'll turn positive. And if that is what we're looking for, if we're looking just for STDs, we're done. We don't know what's there, but we know it's not gonorrhea, and that's what they asked me to test for. So that is a real quick, real easy. The other one is Pseudomonas versus the Enterics. Pseudomonas is oxidase positive. All of the Enterics are oxidase negative. Urease determines whether an organism can hydrolyze urea. Christensen's urea is preferred. Medium is an auger slant. The organism is just streaked onto the slant, not stabbed on this one. It has phenol red. Hydrolysis of urea produces a bright pink color. The whole media will turn bright pink. This is actually used testing for an organism that I had and had to get treated for, called H. pylori. Helicobacter pylori causes number one cause of stomach ulcers, also can cause stomach cancer. It's urea positive. So when you're looking for that particular organism, if you put it into urea and look at it in 24 hours, it will be hot pink. The color of saffron, color of Kennedy's jacket. I mean, it's a bright, dark pink. They can actually do this with stomach biopsies. They have a media card that has a little dime-sized piece of urea media in it. And they'll go in when they scope you, and if they see an ulcer, they can like grab a little piece of it, a little biopsy, a tiny, tiny little piece, and put it on that, seal it down, send it to micro. We look at it in 24 hours, and if any H. pylori is there, it'll be bright pink. And of course, there are other organisms that are urease positive. That's the one I'm most familiar with because we do a lot of testing for H. pylori. Because anyone with stomach ulcers, we're concerned if we don't get rid of that, it will cause stomach cancer. Like I said, a lot of people have ulcers, and this is the most common cause. Difficult to kill too, because it takes several months of antibiotics. I had to do it. Yeah.
Okay, so hemolysis is destruction of red blood cells. What in this paragraph urease, we've got hydrolysis.
Hydrolysis.
What's that the destruction of?
Hydrolyzing urea, breaking urea bonds. Hydrolysis, hemolysis, you're still talking about destruction.
Hemo meaning blood and hydro meaning
Water-based bond. Urea is a big molecule, you know. So here's that little test card that they do the stomach biopsies on. It's called the Clo test, but it's nothing in the world but a urea test. You can see the color it changes when it's positive. And like I said, in reality, I mean, this is blown up real big. That card is this big. About half as wide as it's about that wide. So half as wide as a credit card. And it has this on it, and you put a little tiny sliver of a stomach biopsy on it, wait for it to turn pink. So it's like one of the simplest tests in micro. It's like negative for H. pylori, done. LIA slant. It's inoculated the same as the TSI, used in conjunction with the TSI, because this has lysine in it. This is one of the things that help us differentiate Salmonella from another organism, Citrobacter. Salmonella is lysine positive. Citrobacter is lysine negative. It's very similar to the TIE because it'll also detect the H2S and all that stuff. It just has that one component that's different. That's a clue we need for our Salmonella IDs. And that's why it was utilized. The MIO auger, motility endoornithine. So it's semi-solid auger used to detect motility, just like the motility auger I mentioned. You stab it in, straight out, and if the line goes fuzzy, we know it can swim. If it turns purple, then we know it can utilize ornithine. And then at the very end of the test, after we read that, we'll add that Kovacs reagent straight to this tube to give us our endo result. So we get 3 results out of this one tube. Sulfide endomotility, or SEM auger, is very, very similar to MIO, except that in this one we're reading for H2S production, indole, and motility. Instead of ornithine, it's H2S. So very similar, except for that one thing. Problem with this one is, if the whole thing turns black, how are we gonna read motility? That's the problem with this thing. That's why it was very rarely utilized. Manual multi-test systems. Uses the standard biochemicals we've talked about, but packaged much more compactly. So one of the ones they used was this one. This is an old one that's dried out, but this has 20 wells on it. 20 different tubes, basically, were on this card. 20 different of those two biochemical tests that we eliminated and put onto one card. Same exact test. I mean, this has ornithine, glucose, xylene, indole, a lot of the same ones we've talked about, but it's miniaturized. Same media, same everything. So the technology here has not changed. The size has changed. One of the automated instruments uses these. This is called the microscan. These wells all contain the exact same biochemical test we just talked about. The organism would be suspended in saline and then like flooded across this to fill the wells. Then this whole thing's put in the machine. We're done with it. We don't even read them manually and go, okay, the ornithine's positive, that's positive. Machine looks at it and spits out the results. But it's using the exact same test.
That's one with.
Again, miniaturized.
That one you don't.
Yep. We'll go get one more, we'll be right back.
This is from chapter 9. Yeah. Where do these numbers at? Oh my god. Oh, never mind. Doesn't make sense.
These are also.
This card here comes from the Vitec machine, which is what we use at National Park.
Print these out.
You see all these little holes in it. Every one of those is a different biochemical. It has this little straw here, because in this one, you would inoculate the organism in saline, make that heavy suspension we talked about. There's a special rack this goes in, and this goes down in the tube of that liquid. You put it in the machine, it pulls a vacuum, which sucks it up and fills these wells. Now this goes in the machine. Every 4 hours, it'll look at it and goes, "It done yet?" Nope. It'll go on around. It'll look at it and go, "Okay, they're all done. Salmonella." So once we load it on the machine, we're done. Then when it's done, instead of having 25, 30 glass tubes to throw away in a sharps container, we got this one little credit card going biohazard because it's plastic. Time-wise, notice almost all these said incubate 24 hours. A lot of times with these automated ones, they're looking at the very first hint of the color change. And they can sometimes give you a result in 4 to 6 hours. So it saved time, it saved a ton of space. Just think about how small an incubator you could have, even if you were reading these manual and put 200 of these in. And every 200 of these, each one is, how many tests? 50? You know, so it's so much more compact, so much faster, so much easier. And that's why we've gone to the automated method. But the reason you still need to know most of this is because these automated methods are still using the same exact test. They're just not in tubes anymore. The first one I showed you, that card, that's the API. Series of 20 cupules that contain the substrates and all that. The ID, the tri-panel, is the same as that API. It was just a multi-card where you could do like 3 different organisms on one card. Think of it about the same though. There's also a tube form of that. You see on the right, the Intero tube. Each one of those wells is a biochemical tube. Exact same technology, exact same test. Different size.
How big are those? Like, is it-
The internal tubes
I mean, like, how much stuff is in that, you know?
and these,
Yeah. Like, it looks like the microwave.
and they're tiny. Those internal tubes, they're about that big around,
Oh,
and they're about that long. And like I said, each one of these compartments is a different biochemical test. So it's got 15 different tests on it, and it's like reading 20 tubes at a time. Instead of having a whole rack full of glass tubes, you got this one little plastic device. Rapid automated systems. The term rapid is very relevant because we're talking about micro. We're still talking about a matter of hours, and often it's still 24 hours. So rapid is relevant. We're not talking like rapid like a UA where you dip it and it takes 2 minutes. So some of the rapid tests, endo-catalase, oxidase, you know most of those. The identification systems relying on carbohydrate utilizations, like that API, like the Vitec, that have all these multitude of tests. Still, we're saying rapid in a term of hours. So it's not rapid like some of our other rapid tests, like the 60-second pregnancy. The microscan was that tray I showed you, that you know has the little tiny micro cups of wells. And if you want to look at this tray before you leave, if you look at it, it's like 50 little tiny plastic test tubes. I mean, they're just little size of a pencil eraser wells. You can also do the antibiotic susceptibility on these. And on those, it's based on a cloudy versus clear, and that's what the machine's reading. If it's cloudy, that means there's still organism there, so the antibiotic didn't kill it. If it's clear, that means the organism couldn't live there, that antibiotic works. So for those, it's not really a color change reaction like most of these, it's a cloudy versus clear. But the same technology will do that for you. The machine will read that for you to let you know which antibiotics work, which ones don't. The Vitec I mentioned, the BD Phoenix is another method. There's one called the Sherlock. These are all automated methods to do the same biochemical test. Evaluation of identification systems. The problem with that is, let's say, okay, we're doing this old outdated methodology. I want to bring in a brand new machine. You can't just switch to that brand new machine. Even if they know it's better than what you're doing, you have to compare it to what you're doing. So for months, in your tubes, you get salmonella. You have to take that organism, put it on this machine, make sure it gets salmonella. All of your QC organisms, of which there's about 30, you have to run on that machine every single day for 40 days. If it misses one time, you got to start over. So even if you try to go to new technology, it's not as easy as just buying that piece of equipment and switching to it. There's still a lot of work involved. So you have to compare the accuracy of it, the speed of it, the cost effectiveness of it. There you see the Vitec, the little card. You scan it in the machine, put it in the machine, it tells you the ID. And remember, we're talking about micro. These automated machines will never tell you this is E. coli. It will tell you this is E. coli with 96% certainty. This is E. coli with 92% certainty. Because even if I go so far to say, like as far as the gram positives we just finished, Staph aureus is coagulase negative, or coagulase positive, sorry. Staph aureus is coagulase positive. There'll be 1 out of 100 that'll be negative. So in micro, there are no certainties, but there are percentages. We know that 97% of Staph aureus are coagulase positive. So if it's positive, we can say with 97% certainty, this is Staph aureus. There are no sure things, and these machines will give you the percentage when it prints that out. And your site will have a criteria as far as where it will not take it. Like if it's below 80. If it says I'm 79% sure, can't turn that one out. gotta do further testing.
How often, whenever even where we did equipment or procedures or whatever change, like, they ended up buying stuff like a different type of reagent or something, or from another company?
Frequent. Frequent. Usually, that's not an issue for the normal tech, that's supervisor problem.
Well, I mean, because I said that having to utilize it.
Which means in chemistry, that was my problem. But like, even if somebody in a different department is bringing in a new machine, if I was over there that day, then I had to do their testing for them or whatever, but they do the evaluating, not me. In chemistry, I had to do it.
Do you ever just look at a vendor and just like, it just, no, I don't.
It's not worth it.
I just don't care at the moment, can you get away from me for?
Frequently, yes. When we brought vitamin B12 into the lab, the way I did it, is at the time we were sending all of our vitamin B12s out. I just kept sending them out. But before I sent them out, I'd run them on my machine. Then when the results came back, I would compare, and that was my comparison set. So I jumped out of order a little bit on this, because the chapter I was supposed to do, the first 2 slides I looked at were mentioning biochemical tests. I'm like, that makes no sense, you haven't even heard of most of these. That's why I jumped on that one first. This is all pre-information basically for chapter 19, which is a biggie, and it's big enough that we're gonna spend at least 2 lectures on that one chapter. But it's gonna talk a lot about these biochemical tests. How do you tell E. coli from salmonella? How do you tell this from that? It's gonna mention them a lot. That's why I wanted to get this out of the way, so you can go, oh yeah, I remember that one.